Aloha everyone and welcome back to Bio 101 at Chaminade University. Today we'll be covering Unit 5 where we discuss the cell membrane structure and function. So today we're going to talk about membranes. Now just like everything in biology, the form of the membrane is related directly to its function, which is to separate the inside of the cell from the external environment. And it's semi-permeable in that certain things are able to come in and other things are not able to pass. So we'll talk about how the membrane allows things to pass or does not allow other things to pass across the membrane. And then we'll talk about specialized junctions that allow cells to connect to one another and also communicate with one another. So we'll end there with different cell junctions. So let's start with the membrane. So as you may remember when we talked about biomolecules, the plasma membrane is comprised of phospholipids. And it is a phospholipid bilayer, meaning that it is two layers of the phospholipids. And that's because phospholipids have two different regions within them. One is going to be hydrophobic and one is hydrophilic. And that allows for the segregating property of the membrane. And we'll talk about that in just a little bit. But it allows, as I just mentioned, the isolating function of membranes is really the important part. It's what keeps the external environment separate from the internal environment of the cell so that the cell can maintain what we love to call homeostasis. And there's specialized proteins that are embedded within the phospholipid bilayer that are responsible for allowing certain things in and not allowing other things through. So exchanging of substances, basically allowing in nutrients and exporting cellular waste, and also allowing for communication with that external environment and control of certain biochemical reactions, which also happen at that plasma membrane. And last but not least, very importantly, in fact, the formation of attachments between one cell and the other cells that surround it. Again, the function of the plasma membrane is isolation of the internal and external environments of the cell. But it's semi-permeable, so it's not just a rock wall, right? It's going to be a gate that allows exchange of certain things in and certain things out. It also allows for the communication between cells, which allows for the cells to be able to relay messages to one another and also to respond to those messages so that the body can act in concert. And it is going to allow cells to make attachments between each other that's going to allow for cell stability and also for the interaction of cells where they can pass information without having to leave into the external environment. And last but not least, it's a site by which biochemical reactions are going to occur. So it's an area whereby we can regulate biochemical reactions that occur both internal and external to the cell. Now, when you think of the plasma membrane, you think of like a plastic baggie or something like that, where it actually is just a solid mass that's going to prevent things going in and out. But that's not exactly true. It's more of a fluid mosaic in which the phospholipids and proteins are all able to flow amongst each other within the lipid layer that they are located. So the internal surface never really interacts with the external surface, but along the entire external surface, all of the proteins that are embedded in that external surface can kind of move at will along the fluid mosaic. So it's kind of like a patchwork of multiple proteins and phospholipids that are going to be shifting and flowing within this viscous fluid, if you will. All right, so just to refresh your memory, if you didn't get the chance to watch the biomolecules lecture, a phospholipid is going to be depicted from here on out as a circle on top with two legs on the bottom. And what are we showing? We're showing a head here, which is a phosphate head, and we're showing two of the lipid tails. And the head region, as you can see depicted as a series of globes all the way down, those are going to interact with the aqueous environment, so they'll interact well with a watery environment. And the reason it's a bilayer is because this region here, which is hydrophobic, is going to interact with the regions that are hydrophobic, so the hydrophobic tails of the bottom layer, which allow for a region that is lipid soluble, right, or hydrophobic, that separates an aqueous environment from another aqueous environment. So these polar heads and those polar heads interact well with water, and these tails are going to interact well with lipids. And that's why the phospholipid is so successful, because it's what we call amphipathic. And amphipathic means that it has one region that is water-loving and one region that is water-fearing. So it allows it to interact with both hydrophobic and hydrophilic molecules. But I digress. Some other things that you're going to find among this phospholipid bilayer include proteins, specialized type of proteins, including recognition proteins that are going to help separate self from non-self, glycoproteins that are going to have carbohydrates that stick off of the protein region, different proteins that are going to allow for things to come in and out, like these pore regions, or we also have different extracellular matrix proteins that are going to bind into and lodge through. We also have cholesterol, which is not a protein, it's actually a steroid, but I digress. And we have here the cytoskeleton, which is going to line the inside of the cell, kind of like a scaffolding. So we have scaffolding both inside in terms of the cytoskeleton and outside in terms of the extracellular matrix. So that's what's going to kind of hold this plasma membrane in place and is going to be the site of exchange from the external environment to the internal environment. All right, so why are phospholipids important? They are amphipathic. Again, amphipathic is a large word that simply means it has two regions that are very different, one that's able to interact with water, so that polar region, which is the hydrophilic head, and one region that is able to interact with lipids, the nonpolar hydrophobic tails. And this is important because both the external environment and the internal environment are going to be an aqueous environment. So the external environment of the plasma membrane is bathed in an interstitial fluid. Now, an interstitial fluid is a fluid that is similar to blood but doesn't have any of the blood cells or the blood proteins. Basically, things are going to emigrate out of the bloodstream through leaks in the capillary walls and going to enter into the interstitial fluid, where they're then going to travel from the interstitial fluid across the membrane into the cell if they are something that the cell needs, right, if it's a nutrient. And at the same time, cellular waste are going to be secreted out of the cytoplasm into the interstitial fluid, from the interstitial fluid into the blood supply, where they can be excreted from the body. So the membrane is the region that separates the interstitial fluid from the internal environment of the cell. All right, as I mentioned, for the purposes of this class, I'm never going to ask you to do any sort of chemical drawings or nomenclature, but I want you to kind of recognize that we have one region that's hydrophilic, that's this head region here, it's a polar head, and two hydrophobic tails. For the rest of this class, you're probably only going to see it as a circle with two lines. But while I have your attention here, let's pay attention to the fact that this tail, which is a fatty acid tail, right, is saturated. That means there are no double bonds to any of the carbons in here. This one is unsaturated. There is one double bond. That means that we could possibly break that double bond and saturate it by adding two more hydrogens. Now, some phospholipids are going to have, both of them are going to be saturated. Both of them are going to be unsaturated. Some of them are going to be a mix of each. And as we may remember when we talked about olive oil and bacon grease, the more saturated something is, the more rigid the structure, and the less saturated, the more fluid the structure, which is why organisms that are found in warm environments have a different phospholipid composition than individuals that are found in colder environments. But I digress. Again, the phospholipid bilayer is the region of the membrane that's considered to be fluid in that fluid mosaic model. And it is going to face both an interior and an exterior environment that is aqueous, or comprised mainly of water. And that external environment, again, the interstitial fluid, the internal environment, again, the cytosol. And the reactions or the interactions that create that phospholipid bilayer are all done mainly driven by hydrophobic and hydrophilic interactions. So the tails are going to automatically orient with the tails the same way that oil in water is going to glob up and find itself on the surface, right? A lot of tiny globs of oil are going to come together and form a large glob of oil. That's the same thing that happens with the phospholipids that create these plasma membranes. Basically, the tails are going to interact with each other and the heads are going to align with each other just by hydrophobic and hydrophilic interactions, and eventually it's going to form a complete sphere, which is going to be the cell membrane. Again, this looks kind of like this. The phospholipid has a head that interacts with the interstitial environment, and a hydrophobic tail that interacts with the hydrophobic tail of the other layer of the plasma membrane, and that's what separates the internal environment, which is the cytosol, and the external environment, which is the interstitial fluid. Both of them are aqueous or comprised mainly of water. But this is oversimplified because it's only showing the phospholipids. Remember, we also have channel proteins and receptor proteins and the cytoskeleton. So there's a lot of other players that are going to be found embedded in this fluid mosaic of phospholipids. The other cool thing about the phospholipid is that it's a flexible membrane, so it can bend in different directions. Even though it's very strong because of those hydrophobic interactions, they can actually kind of slide along each other, so it's fluid. That allows for the shape of the cell to change, particularly in an animal cell, because the individual phospholipids are not actually bound to one another, so they can kind of move however they choose within that layer that they've been bound to, or that they've been stuck within. Now again, some of them are unsaturated and some of them are saturated, so the tails are going to be a little bit different in terms of their composition, and that allows them to be slightly slippery, which makes it more of a fluid environment. So that means that these guys are actually able to slide across each other fairly easily. As I mentioned previously when we talked about biomolecules, the more unsaturated, the greater the fluidity, the more saturated, the less fluidity. And we talked about this previously being similar to a bunch of paper clips that were folded flat. You could fit more flat paper clips in a box. Then if the paper clips had kinks in them, you could fit less of them in the box. And so that allows for more motion because each of them takes up a little bit more room. So you end up with greater fluidity for the unsaturated tails than you do with the fully saturated tails. And that's going to be the difference in the phospholipid composition of organisms that live in colder environments, right? So they need a little bit greater fluidity, or in warmer environments where they need a little bit less fluidity. Now, the role of a phospholipid bilayer is not just segregation, right? It's semi-permeable, and that means that certain things are going to be allowed in while other things are not. So certain things are able to cross very easily, like water, but other things are not able to cross very easily. Things like salts, amino acids, and sugars are going to have to be transported in to the membrane. So the isolation, as I mentioned, it's semi-permeable. It's not complete. So certain things are able to pass through. You can picture this, the difference between like a chain link fence and a brick wall, right? Small organisms could pass through the chain link fence when they couldn't through a brick wall. So small molecules like water and oxygen and carbon dioxide are able to slide right on through there, as well as things that are a little bit larger but are lipid soluble. So remember, the lipid soluble environment is something that it would be favored inside that plasma membrane area. So lipid soluble molecules are going to more easily be able to pass through that selective barrier than ones that are water soluble. Now, because membranes are susceptible to temperature changes, they become more fluid at high temperatures and less fluid at low temperatures, and so that's why I mentioned that we tend to have more of the unsaturated, that means they have more kinks in them, in organisms that live at low temperatures than organisms that live at high temperatures. But an additional thing that helps stabilize that membrane is cholesterol. So cholesterol, which is a steroid, right? It's got sterol right in it. Is going to be a membrane stabilization component and helps to reduce permeability of the plasma membrane. So it helps to affect the fluidity by reducing fluidity, which helps also reduce permeability because nothing goes through the cholesterol molecules the way that they do through the phospholipids. Now, again, it's not just a layer of phospholipids. There are a ton of other things embedded within there, such as proteins. Some proteins go all the way through, right? They're transmembrane. Some are just embedded in the membrane, right? Embedded in one side or embedded in the other side of the phospholipid bilayer. So maybe they're only embedded in one side and don't go all the way through. Some of those proteins have specialized carbohydrate regions. These are called glycoproteins, and those are going to be kind of identification proteins. But in general, membrane proteins can be grouped into 5 major categories. Now, this doesn't mean that there's 5 proteins, right? There's a ton of enzymes, there's a ton of recognition proteins, but there are 5 major roles that these proteins play in membrane function. And the first is enzymes. And enzymes are going to get their own chapter entirely, but enzymes are specialized form of proteins that speed up the rate of biological reactions. They're biological catalysts, and they are very specific to the reaction that they catalyze, but they are going to be very important on the cell surface and also on the inside of the cell to be able to help build things up and break things down. Additionally, we have recognition proteins. Now, recognition proteins are going to be proteins that are identifying markers that say, hi, I'm a heart cell, or hi, I'm an eye cell. But more than that, right? I am a pig heart cell, or I am a human heart cell, or I am Dr. Griffiths' human heart cell. So they're very specific, these recognition proteins, and they're actually something that people who end up with heart transplants have to kind of overcome, but because the recognition proteins on the transplanted organ would not be the same as the individual's recognition proteins, so they have to take immunorejection drugs to prevent the body from attacking those donor organs. But I digress. We also have receptor proteins. Receptor proteins are going to be specific to a ligand. So basically, a receptor is bound to the membrane and a ligand is free in solution, but a receptor and a ligand are going to interact to cause some sort of cellular change. So you can consider this some sort of messenger molecule, right? This is a messenger relay system, whereas when the messenger interacts with, the messenger being the ligand, interacts with the receptor, something triggers an alarm and passes information into the cell somehow. And it usually does this by way of a conformational change, some sort of cell signaling pathway that goes into the cell and changes the inside of the cell metabolism somehow. We also have connection proteins, which are involved in basically holding one cell to the next cell or to the basal membrane, basically what helps keep all of your cells from dissociating at any time. So if you were to pinch your skin, for example, and pull a little bit, you're not going to rip all of your skin cells apart, and that's because your skin cells are connected or tethered to the skin cells next to each other by these connection proteins. And last but not least, they are actually highly important. We have transport proteins, which are responsible for moving other things from one side of the cell to the other. So we're bringing in something that we would like to bring into the cell or exporting out something that we would like to release from the cell. All right, so now that I've told you what I'm going to tell you, I'm going to go ahead and tell you the same thing. Enzymes are specialized proteins that promote chemical reactions. They increase the rate of a reaction by catalyzing a reaction that would already happen, but increasing the rate at which it would happen by sometimes 10,000-fold. So basically, they synthesize or break apart biological molecules. So they promote anabolic or catabolic biological reactions, and the sum of all these enzymatic activities and biological reactions is metabolism. And these are very specialized plasma membrane enzymes that can create, remember every enzyme has a specialized function, it only does one thing. And these enzymes are helpful to help create the extracellular matrix. The extracellular matrix is what is surrounding that interstitial fluid. It's basically all the proteins and glycoprotein fibers that fill in all of the spaces that surround the animal cells. And as we age, the loss of the extracellular matrix is what causes things like wrinkles and crow's feet. We still have the same amount of cells, but the area that connects the cells gets very lax and soft and does not fill in the same way that it used to. We also have receptor proteins, which, as I mentioned, are going to cause an intracellular cascade or some sort of cellular responses. Receptors bind to ligands. So the specific molecules that bind to a receptor is a very specific lock and key interaction where the ligand, which is free in solution or floating through the bloodstream, then ends up binding to the receptor protein on the surface of the cell, right? So ligand could be a hormone, for example, or epinephrine or something like that, sent from a different cell through circulation to trigger a response within the cell. So you can picture these guys like the Paul Revere. They're basically picking up information from the external environment and passing that information on to the internal environment to alert the cell that the British are coming, for example. Recognition proteins are any identifying markers. They are glycoproteins generally, so that means that they have a sugar on the outside of them, and they're going to be identification tags. Again, this is what's going to differentiate self from non-self, so, and even within the body is specific types of cells. So not just that it's a human cell, it's Dr. Griffiths' cell. Not just that it's Dr. Griffiths' human cell, but it's actually Dr. Griffiths' human cell that's for the eye, or something like that. So recognition proteins are very specific that identify the cell to anything that might be traveling through solution, like antibodies that are part of the immune system, for example. All right, so this is what receptor activation looks like. So here's our receptor protein, and it's very oversimplified, but in this version, when the receptor is inactivated, it has a square bottom. Okay? Now a ligand or a messenger molecule of some sort comes through. It could be a hormone, it could be epinephrine, it could be cyclic AMP, but some sort of messenger molecule binds to this receptor protein. Now mind you, every receptor protein has a specialized pocket, so not every messenger molecule fits. Only specialized messenger molecules fit with the specialized receptors, but once the specialized messenger and receptor combination is created, we have a change on the inside of the cell. So the binding happens on the outside of the cell. The ligand or the messenger molecule never comes into the cell. But this transmembrane receptor protein has a conformational or shape change that occurs at the inside of the cell that sends a message or a relay. Generally, it's going to cause an intracellular cascade, so A is going to get turned into B, into C, into D, but is eventually going to cause a biological change within the cell, some sort of response within the cell. Now, connection proteins are probably the easiest to understand. Connection proteins are anchoring proteins. They anchor cell membrane of cell A to cell membrane of cell B, or perhaps cell membrane of cell A to a basement membrane of some sort. But connection proteins are just proteins that are linkage proteins and form connections between adjacent cells. So they basically are going to link the cytoskeleton, which is inside the cell, to the extracellular matrix, or to another cell's cytoskeleton. And we have a couple different types of connection proteins, and we'll talk about them in just a minute, but we're going through the overview right now. We also have transport proteins. Transport proteins are involved in bringing things in and sending things out. Remember, not everything is going to be able to travel through the plasma membrane directly. It's semi-permeable. That means that some things have to come in through specific channels. So we have channel proteins that are more like pores. Again, you can picture a channel just opening up. Anything that is going to fit through it is going to be allowed through. So it's a specialized specific substance that when the channel is open, that substance is going to be allowed through until the channel is closed. Now, a carrier protein is going to bind something that's specific. So both channel proteins and carrier proteins are specific to the substances that they bind, but the carrier proteins bind substances and bring them through the membrane one at a time. Sometimes this uses cellular energy and sometimes it does not use cellular energy, and that depends on the concentration of whatever it is that we're talking about on the inside and the outside of the membrane. Okay, so when we talk about things that are in solution, right? We talk about the cytosol or we talk about the interstitial fluid. We want to talk about the concentration of something in solution. And in order to talk about concentration gradients, we have to talk about solutions themselves. And solutions are created of a solute and a solvent. So a solvent is whatever the fluid is that is going to be what the solute is dissolved in. So if, for example, you add sugar to water, sugar is the solute and water is the solvent. And water is described as the universal solvent because most biomolecules are going to have no trouble dissolving in a water solution. Now, if we talk about the amount of the substance that is in that solvent, now we're talking about concentration. So if I put 1 tablespoon in a glass of water and in a different glass of water of the same volume, I add 2 tablespoons, well, now I have double the concentration in the second glass, right? So the concentration is basically the amount of the substance inside the amount of the given solvent. And if we have a difference in these two, then we have a concentration gradient. Now, a gradient can be from any physical difference: temperature, pressure, charge, or concentration. So gradient simply means that there's a difference between two different solutions. And so, for example, if it's warm on one side of the house and cold on the other side of the house because you have the AC running, then you have a temperature gradient throughout the house. And probably somewhere in the middle of the house, you have a medium temperature, right? So a concentration gradient is basically when we have a high concentration of whatever it is on one side of the cell and a lower concentration on the other side of the cell. And the cells need to maintain homeostasis. So what that means is, irrelevant of what is on the outside of the cell, you need to have X amount of whatever it is that you're talking about, whatever your substance is. So if there's not enough on the outside and you need more of it, you're still going to need to bring it in. If there's a plethora on the outside and you have too much of it, you're still going to need to send it back out. So we have to use transport proteins to maintain these concentration gradients, right? The differences in the solute concentrations on the inside and the outside of the membrane, because the inside of the cell can only have a certain parameters of whatever our substance is. Now, the process of diffusion is when whatever your solute is going to go from a higher concentration to a lower concentration. And basically what that means is that if you put a drop of dye into a glass of water, at first, the drop of dye is going to be like a little teeny tiny dollop of blue, and the rest will be clear. But then over time, that dye is going to diffuse out to make the entire glass of water light blue with no regions that are going to be dark or light, and that's called equilibrium. So gradients are going to be a way by which molecules are moving from one place to another. They usually move along the concentration gradient. So again, they move from high to low, whether it's a concentration or a pressure gradients. Basically, that's going to move molecules or ions from high to low to reach what we call equilibrium, anytime we equalize the difference. And if we have to go against that concentration gradient, we're going to have to use energy and specific cell membrane proteins to bring things in or out depending on what concentration we have of that molecule or ion on the inside or the outside of the cell. Now, if you picture that molecules and ions are constantly bouncing off of one another, and the rate of that motion increases as we increase temperature. Picture boiling water is going to move a lot more than cool water, right? And so as we increase the temperature, the molecules of water are going to bounce off of each other more. And that's the same thing with molecules and ions that are in solution. They're moving around more, which is why if you increase the temperature on something, you can get more of whatever your solute is to dissolve in the solvent. So for example, if you can super saturate a solution by heating up water and adding more and more and more sugar to it, and then once it cools down, you get something like rock candy where the sugar crystallizes back out of solution. And so the increase in temperature increases the rate of the motion inside whatever the solution is. And that in and of itself, that random motion is going to produce equilibrium eventually, right? Where things that are going from the high concentration to a low concentration until they eventually are going to get to the point where they have equilibrium, which is when we have the same amount of whatever the substance is throughout the entire volume. So the summary of diffusion is that diffusion is the net movement of whatever our molecule is from a high concentration to a low concentration. And if the concentration gradient is higher, the rate will be faster. So if we have 20% versus 80%, then it's going to be a lot faster than if we have 20% versus 30%, because there's a much greater difference between 20 and 80 than 20 and 30. So if we have two different solutions that have a much larger concentration gradient, like a greater difference between the two, and we put them together, the rate of diffusion will be much faster. The same thing occurs with temperature. The increase in temperature increases the rate of diffusion as well. And if we have no intervention, eventually diffusion occurs until we reach equilibrium, that is, until the concentration gradient has been entirely eliminated. And this is the drop of dye example that I was just talking about. If we put a drop of dye in a glass of water and allow for the random motion to occur, eventually the water molecules diffuse out into the dye until we reach equilibrium. And this will happen faster if we have a greater temperature, for example, or a greater concentration gradient. But what's the difference between that and a cell? The cell has a plasma membrane, right? So the plasma membrane is selectively permeable and allows only certain things through, and that's going to help maintain a difference from the inside and the outside of the cell. But water is able to go in and out kind of at will, and we'll talk about how important that is, maintaining the same amount of water on the inside and the outside of the cell, in just a little bit. But let's talk about motion across the plasma membrane. So some things are going to go with the concentration gradient. That means that there's a larger amount on the outside of the cell than the inside of the cell, and we want to bring it in. Or there's a larger amount on the inside of the cell than the outside of the cell, and we want to send it out. Right? If we're going from high to low in the same direction, then we can just open up a floodgate and allow it to leave, right? That's passive transport. And energy requiring transport is any time that we require the use of ATP or cellular energy, and usually that's when we are going across the concentration gradient. And the example I like to use here is if you are in a boat that has a small hole in it and you're sitting on a surface of water. Allowing the water to come in, right, there's more water outside than inside, allowing the water to come in with that concentration gradient along it is no problem, right? It's a no-brainer. That water is going to come in with no energy. But if you want to get that water out against the concentration gradient, still we have more water outside than inside, but you don't want it to come in, you want it to go back out, then we're going to have to bail that water out, and that's going to require energy, right? So energy requiring transport is any time that we're going against the gradient, it's going to cost us cellular energy. All right, I'm going to skip that because that's basically telling you what I'm going to tell you. That's a summary of everything that I'm about to talk about. So passive transport has three major types. One is simple diffusion, one is facilitated diffusion, and the other is osmosis, which is simple diffusion specifically of water. And as we talked previously about diffusion, diffusion is basically just going from, we had originally high and low, so we had a concentration gradient, and then eventually we're going to end up reaching equilibrium. And that's going to happen across a fluid, or within a fluid, or across a membrane, if the membrane is permeable to whatever that substance is. So simple diffusion is pretty easy. Basically, molecules are going to go directly through the phospholipid bilayer, right? Go straight through the membrane because they're small, or they are lipid soluble. And they're going to go from concentration of high concentration to low concentration. So certain things are just going to come right on in, including things like water, which sounds counterintuitive because water isn't lipid soluble, but it's small enough that it slides right on through. It basically hops from one of the polar heads to the other polar head and swings on through. We also have oxygen, carbon dioxide, and other lipid soluble molecules that can get a little bit larger, like alcohol, and vitamins A, D, and E are all going to be able to go across the membrane by simple diffusion. And simple diffusion is affected by multiple factors, including temperature, molecular size, and the solubility in lipids, right? The more soluble you are in lipids, the larger you can be and still slide through that plasma membrane. All right, so we can have multiple different types of diffusion. Here's simple diffusion. Simple diffusion is something like oxygen. Oxygen is going to go right on through the plasma membrane in between two phospholipids and sneak right on. But it's going to go from concentration of higher on the outside to lower on the inside. This is also going to count for carbon dioxide, which because cells are exporting carbon dioxide, right? It's a byproduct of cellular metabolism. We're going to have more carbon dioxide on the inside of the cell than on the outside. So it's going to go across the same way from a high concentration to a low concentration. So oxygen and carbon dioxide are going to slide right across that plasma membrane without costing any cellular energy. But if we want to have a carrier protein or a channel protein, these are two mechanisms by which we can take something from the external environment and bring it into the internal environment. Again, we are still going with the concentration gradient. We are not costing any energy here. So we have a higher glucose on the outside than we do on the inside. Specialized carrier proteins can pick up only one molecule at a time. They have a conformational change where they release that molecule and then they revert back to their original state. So this can pick up one glucose, pop it in, then start over, pick up another glucose and pop it in. That's what a carrier protein does. It has a specialized pocket that can carry a larger biomolecule. Now a channel protein, picture that this has a gate on it. Channel proteins can open and close. Channel proteins are going to allow as many of whatever the ion is, so ions are usually a lot smaller than some of these larger biomolecules, straight on through this pore basically. So it's going to be a way of diffusing again from high to low, but it's facilitated because it's helped by these channel proteins or also by the carrier proteins here. And then last but not least, we have osmosis. And osmosis is selective. And it is still going from high concentration to low concentration, but it is going by way of either straight through the plasma membrane or through aquaporins, and we are only talking about the diffusion of water. So osmosis is very specialized to the diffusion of water, which is why when someone says they're going to sleep with their biology textbook and they're going to learn the information by osmosis, not only is it ridiculous, but it's also using faulty vocabulary. All right, so facilitated diffusion is basically when we are transporting things across the membrane. We're doing that either by the carrier proteins or by a channel protein. So we have specialized transport proteins that are moving these molecules across. And if we use specific things that are water soluble, like ions and amino acids, they're able to go down across their concentration gradient. So same thing, high to low, using the channel and carrier transport protein system. So it's a facilitated diffusion. We're not using any energy. We're still going from high concentration to low concentration, but these are larger biomolecules that can't just fit through the chain link fence. They have to go through specific gatekeepers, and the gatekeepers are specific for each biomolecule. So again, we have carrier proteins that have specific regions that bind that specific molecule, and then they undergo a conformational change, so they bind it on the outside, and they bring it inside, and then they release it and lather, rinse, repeat, or vice versa. They bind it from the inside because we're trying to export it, and then they release it to the exterior environment. But either way, they're going in a concentration gradient, so they're going from high to low. Again, we're not costing any cellular energy. Another type of facilitated diffusion protein is going to be, or transport protein is going to be a channel protein. Channel proteins have pores, and they have a little door basically that opens and closes to allow things in or out, but again with the concentration gradient, so from high to low. So it's going to allow them from one side of the membrane to the other, but it's not going to cost us any cellular energy. And if we have specialized diffusion that goes across water channel proteins, those are called aquaporins. So when I just talked about channel proteins, we have specialized channel proteins for every biomolecule. We also have specialized channel proteins for water called aquaporin, which are selective for water based on size and charge, so allowing only water molecules to travel through these little specialized channel proteins, kind of like VIP channels. Now, osmosis, as we just mentioned, is the diffusion of water. It can go either through the aquaporins or through the membrane itself. And the permeability of the membrane to any of these ions, but also water, is specifically able to be changed in response to certain things. So it's influenced by the concentration gradient, for example, the pressure gradient, for example, the temperature gradient, but very importantly, the water gradient. So if we are going to kind of flip this on our head, for example, and we picture a sugar solution, that's 5% sugar. Well, if you turn that around, that means it's 95% water. So water is also going to travel across the membrane from a high concentration to a low concentration. So when we are talking about osmosis, I want you to stop thinking about the concentration of solute and instead flip it the reverse of 100. So if we say 5% solute, then 95% solvent, 95% water, right? And so you look at the concentration of water, and water is also going to go from high to low concentration across the membrane. And again, when we figure out the concentration of water, we're going to subtract out the concentration of the dissolved substances. Everything that's dissolved in it reduces the concentration of the water molecules. So for every percent that we add sugar, we subtract a percentage of water, because in all, it always has to equal 100. And Basically, what we are looking at when we look at something called an osmotic strength. Osmotic strength is the tendency of the solution to attract the water across the membrane, and that really has to do with the concentration gradient of water on one side of the membrane and the other. And the greater the concentration difference, the greater the osmotic strength. So what does that look like? All right, so I'm going to introduce a couple more terms here that we haven't talked about yet. I'm going to start with isotonic because it's the easiest to explain. So if we have the exact same concentration of water on the outside of solution as we do on the inside of the cell, then we have an isotonic solution. So what we have, say we have 95% water inside the cell and 95% water outside the cell. Does that mean no water moves from inside to outside? Not at all, but it means that an equal amount of water moves inside and outside, so we have no net flow of water. That means that the cell is going to maintain its regular conditions, right? This is called isotonic or equal tonicity. Now, if we have more free water on the inside of the cell than we do on the outside of the cell, that means we're in a solution that's hypertonic, right? And that means that the water, so say we have 95% water on this side and only 90% water on that side. Now the flip way of saying this would be that we had 5% sugar and 10% sugar. So if you get asked a question, I want you to automatically, when we're talking about osmotic strength, I want you to automatically flip it into the percentage of free water, which again is the equal and opposite number to add up to 100. So if I say 5, it's 95. If I say 10, it's 90. But my point is that if we have more free water on the inside of this cell than we do on the outside, because we have a higher solute concentration out here, then water is going to flow out, right? We're going to end up with this cell shrinking. So a sac that's in a hypertonic solution is going to purge, which means it's going to shrink down. And the equal and opposite event can happen if we have a sac in a hypotonic solution. So what that means is that we have a higher concentration of water molecules on the outside than we do on the inside. Again, that might look like, in terms of a question, that we're saying that we have, say, a 1% solution and a 5% solution. Basically, when you're looking at free molecules of water, you're going to separate that out and make that, say, 99 and 95, right? So 99 is a higher percentage of water than 95. That means water is going to flow this direction into a cell, which means that a cell that's in a hypotonic solution is going to swell up like a hippo, okay? If it's in a hypertonic solution, it's going to purge and get smaller. If it's in a hypotonic solution, it's going to swell up like a hippo and eventually burst. And that's going to be something that's like a lysis. So if, for example, we had red blood cells in that condition, then the red blood cells would lyse open. Again, isotonic, so I just told you, but I'm going to tell you again. Isotonic means that we have an equal concentration of water and also of dissolved substances, which means that although water is traveling in and out, we have no net motion of water in or out across the membrane. And a hypertonic solution means we have a greater solute concentration, which means we have a lower water concentration, which means water is going to move again from high to low, which means that water is going to move out of the cell into the hypertonic solution. That cell is going to purge and get smaller. If a cell is in a hypotonic solution, that's a solution that has a lower solute concentration. And what I want you to read there is a higher water concentration. And again, water is going to go from high to low. So it's going to go away from the hypotonic solution into the cell, which is going to cause the cell to lyse. So to summarize what I just discussed, basically osmosis is how water is going to diffuse across a semi-permeable membrane, either by simple diffusion, which we just talked about it being able to slip right on through the chain-link fence, or facilitated diffusion through channel proteins called aquaporins. And it's going to move the same as anything else down the concentration gradient. So from high concentration of free water to the side with lower concentration of free water molecules. So again, anytime I give you a concentration of solute, I want you to immediately turn it around into solvent. So if I say it's 5% sugar, I want you to immediately turn around and say then it's 95% water. And then you're looking at the high to low from the water concentration when you're looking at osmotic principles. Again, anything that's a solute in the solution is going to reduce the concentration of free water. So every 5% sugar is going to reduce the concentration by 5%. And if we have a semi-permeable membrane and we have solutions that are on one side and the other, if it's isotonic, it will not change. We will have water going in and out at the same rate. If the solution has a higher solute concentration, then it's going to be called hypertonic. If the solution has a lower solute concentration, it's hypotonic. Hyper being higher, picture a child that's hyper on sugar has a higher energy content. Hypo being low, like if you feel like you're fat and lazy like a hippo, you're not going to get as much done. So you have less energy when you're in hippo mode than when you're in hyper mode, right? So hyper is higher, hypo is lower. And just as I mentioned when we talked about the cell in that beaker of water, the same thing is illustrated when we talk about red blood cells. So when red blood cells are put in an isotonic solution, the same net flow of water in as out, we're going to maintain this nice little dimpled shape. If they're put in a hypertonic solution, that means that they have more water inside the cell than they do outside of the cell. So water is going to flow outside of the cell, which means that the cell is going to shrivel. So if it's in a hypertonic solution, it is going to purge, purge. If it's in a hypotonic solution, it's going to swell up again like a hippo. Water is going to have a higher concentration on the outside than on the inside, so more water is going to be going inside the cell and less water going outside of the cell, and eventually these cells here are going to lyse or burst open like a balloon that has too much air in them. All right, so if cells are placed in a hypotonic solution, like 100% water, for example, they're going to swell, and that's because water is going to go into the cell. And again, cells that are in isotonic solutions are unaffected, and cells that are in hypertonic solutions are going to purge and shrink. All right, so these rules and regulations of water and osmotic pressure are very important when it comes to biological systems, right? For example, water is going to flow into the cytoplasm of a plant because it's more concentrated in the extracellular fluid than it is inside the cell. And the same thing is true when we're talking about the concentration of water inside the cell and then the water flowing into the vacuole, right? So the concentration is going to be more concentrated inside the cytoplasm, and it's going to flow into the vacuole. And when we have water inside the vacuole, that's going to cause turgor pressure, or pressure on the inside of the vacuole, which then puts pressure on the organelles, which then puts pressure on the inside of the cell, which is what plants actually use to stand upright. And that's what this looks like, turgor pressure in a plant. When a plant that's very healthy and has a nice normal watery environment is going to have water inside every single cell inside the central vacuole. And that's going to fill that central vacuole, pushing the cytoplasm against that cell wall, which helps maintain the cell's shape. Right? Now, if we don't have enough water, right, the plant is starting to wilt. You can see these leaves are hanging down. That's because they don't have any turgor pressure, and that's because inside each of the cells, the vacuole has shrunken. And when it shrinks, sometimes the cell membrane actually pulls back from the cell wall, and that forms like a weak spot where this can fold over, where the cell wall is entirely unsupported. And when the cell wall is unsupported, the plant is going to wilt. And this process actually is reversible very quickly. If you were to pour a glass of water in here within about 10 minutes, the plant will return back to normal turgor pressure. All right, so again, osmosis is really important in all biological systems, and water is going to be constantly coming in from the external environment if we put something in fresh water. All right, so why else do we need water, right? So water is going to be really important in all biological systems, and some of those systems are going to include energy requiring systems like active transport. Right, I'm going to digress here a little bit. We just previously talked about passive transport, which was the process of biological molecules going along the concentration gradient from high to low into or out of the cell, right? Now, active transport is going to go against the concentration gradient. So if you remember the example where we were talking about being in a boat that had a hole in the bottom, and if we allowed the water to come in, that would be passive transport. It would take us no energy. It would just be going from high concentration of water to low concentration of water. But if we wanted to get the water out of the boat, that would require energy, and that's what active transport is. Active transport is going against the concentration gradient. It's exporting something out when you already have less of it inside than outside, or exporting something in when you already have more of it inside than outside. And we can do this by a couple different ways, right? We can do it by active transport or endocytosis or exocytosis. So endocytosis and exocytosis are vesicular transport. Again, endo being enter, cyto being the cell. So entering the cell by a vesicle or exiting the cell by a vesicle would be endocytosis, or exocytosis would be exiting. And all of the cellular activities rely on bringing these certain things in. So we can do that by bringing in large molecules through endocytosis and exocytosis. And this is vital because we need to maintain homeostasis. And in order to sustain life, we have to maintain these biological parameters. And that means that if we don't have enough on the outside and we still need more on the inside, we're going to bring in as much as we can, even if it's against the concentration gradient, so that we can maintain homeostasis. So this is crucial to maintaining concentration gradients, bringing food in, secreting waste, and communication from one cell to the other. These biological gradients drive all of these metabolic processes. Okay, so during active transport, membrane proteins are going to be moving things against their concentration gradient. Again, we are going, in this case, from low to high. Usually active transport membranes are going to be transmembrane proteins, meaning that they span the entire plasma membrane from inside to outside. And usually because they require energy, they're going to have an ATP binding site. So ATP is basically our cellular dollar bills or $5 bills. And in order to have a process that needs energy, we're going to have to use ATP to do it. And we do that again by breaking down the bond of that third phosphate of ATP. So we go, TP stands for triphosphate, right? We break down TP into DP. So ATP or triphosphate gets broken down into ADP or diphosphate, and that releases energy that was held in that third phosphate bond. And that stored energy is used to drive a molecule against a concentration gradient. That's the energy that they're going to get to pump the water out of that sinking boat, right? And so active transport proteins are actually referred to as pumps, which kind of makes sense because, again, we're going against a concentration gradient. Now, here is an example of a calcium pump. All right, so here we have a binding site. It's going to be specific for calcium. Before I start, let me point out here that we have more calcium on the outside than we do on the inside, and yet we still want it to go out, so we want to go from low to high, so we want to go against a concentration gradient. All right, so we have calcium that binds in the binding site. Additionally, we have ATP. This is like putting our dollar bill in the vending machine. That ATP has to bind to that site as well. And then as soon as ATP is converted into ADP and free phosphate, so we cleave that third phosphate off and we now make a diphosphate molecule, that's a low energy form, that releases energy. And that energy is used to drive the ion, in this case calcium, across the membrane. All right, so that's an example of how active transport works. Another way that we can bring things in and export things out of the cell are going to be vesicular exchange. And this is for large molecules. Anything that's external that is too large to move directly across the membrane is going to have to get engulfed in a vesicle. And it happens by a process, again, called endocytosis, so entering the cell. Exocytosis is the opposite of that, or exiting the cell. And there's multiple types of endocytosis. The first is called penocytosis. It's a little bit smaller than some of the other types of endocytosis. Penocytosis is cell drinking. Basically, we're moving liquids, but also these liquids have different solvent or solutes inside of them. So we're bringing certain molecules or ions into the cell along with these liquids by a process called penocytosis, which literally translates into cell drinking. So we're taking small little sips, basically, cell sipping. The bigger brother of that is called phagocytosis, or cell eating, and that's where we're moving large particles, solid particles, into the cell. And we're doing it in a very similar manner, but kind of on a larger scale as penocytosis. And last but not least, we have a specialized type of endocytosis that's receptor-mediated. And what that means is that we have receptors that are going to be specific for whatever it is that we want to pull in, or waiting for ligands to bind. And then once these molecules bind, we're actually going to, instead of releasing them or causing any sort of intracellular change, in this case, we're going to actually bring those molecules into the cell by creating a vesicle around them and popping that vesicle internally inside the cell. And that's called receptor-mediated endocytosis. But let me show you what I mean. So this is penocytosis. Penocytosis is going to be very similar to phagocytosis, except we're bringing in mainly liquid and not going to be bringing in a lot of solid matter. But basically, we're going to have an invagination, which then is going to bud off even more until finally the plasma membrane touches, and we have a vesicle containing interstitial fluid, which is then going to come inside the cytosol and release its contents. Generally, this vesicle is going to meet up with something like a lysosome, so it's not going to go directly into the cytosol. Sometimes it will release it, but oftentimes it'll meet up with something like a lysosome so that it can be determined if what is brought in is going to be good for the cell, or if we need to break it down into its components, or if we need to destroy it or put it into cellular waste, etc. So this is what penocytosis looks like in a transmission electron microscope. So this is the first invagination. You can see that invagination gets more of a bulbous form, and we have the membranes are coming together, and then eventually they pinch off and come into the cell. Receptor-mediated endocytosis is a little bit more complicated. So these here are regions on the membrane that are going to be causing the invagination in a little bit. So this is a precursor to the invaginated region. Here's the receptors that are on the outside. This is a coated pit. It's coated with what's called clathrin molecules. Clathrin molecules are each going to have a little bit of a curve, and when we have a lot of them together, we're going to make a circle. So this clathrin coated pit is going to have receptors inside where the ligands, in this case some sort of nutrient, are going to bind to that receptor and then be held. So these little tennis balls are going to be held by these little Ys and not let go until the clathrin coated pit is going to, more and more clathrin is going to end up circling it around until we end up with a coated vesicle, the outside with clathrin, that eventually is going to get pulled inside the cytosol. And then eventually that clathrin is going to get released and go back to another precursor region. And that's going to bring inside of the cell whatever nutrient molecules we wanted to by way of this endocytosis. So that's receptor-mediated endocytosis. This is what it looks like on a transmission micrograph. Okay, so here's clathrin coating the inside. Here's our receptors coating the outside. Over time, those receptors are going to get filled and bound by their ligands. At the same time that we get more and more clathrin on the outside, clathrin becomes closer and closer to a circle until it finally is going to pinch off, whereby we're going to have a coated vesicle. So it's clathrin coated, and the inside is going to contain whatever nutrient molecule we wanted to bring inside the cell. Again, this vesicle is probably going to meet up with the lysosome before it gets released into the cytosol, just so we can make sure that we have what we want, and to break it down into whatever its basic biomolecules that we can then recycle inside the cell. All right, so phagocytosis is a little bit larger than pinocytosis. So what we saw previously with pinocytosis was bringing in fluid that might have solutes in it. Here we're going to be bringing in an entire external organism. Here's a fungal cell being engulfed by a white blood cell. Here's an amoeba that's engulfing a paramecium. So an amoeba you think of as small, but paramecium are even smaller. And phagocytosis is different from pinocytosis in that it's going to bring in a full food particle. So we have these little pseudopods that are going to stretch out. So instead of having an invagination on the surface, we have regions that are basically like little, well, pseudopod regions that are going to wrap around that food vacuole and then ingest it. And then again, this ingested food vacuole is going to get sent to the lysosome, where it can be decided upon, where its fate of all of the biomolecules can be decided upon. Exocytosis is the opposite of endocytosis. So exocytosis is where we are secreting undigested particles or waste, and we're doing it by vesicular transport. So vesicles with whatever material we want to secrete are going to be created inside the cell and then bind to the cell surface internally. They're going to fuse with the cell membrane and then release the contents out into the interstitial fluid. And that looks like this. Again, it looks almost exactly like pinocytosis, but it's equal and opposite. So we have a vesicle that is coming from the cytosol that contains within it whatever we want to secrete. It's then going to bud and touch the internal layer of the plasma membrane. Then it's going to open up and release all of the secreted material into the external environment. And this is what it looks like in an actual transmission electron micrograph. So this is all the secreted material being spewed out into the external environment, into the interstitial fluid. Okay, so the reason that cells have to why we end up with multicellular organisms in the first place, why cells have a limit on their cell size, and that is because diffusion is very important, right? Diffusion of biomolecules into and out of the cell is actually relatively slow in terms of biological processes, and it's vital in terms of bringing in nutrients and exporting cellular waste. And if a cell gets extraordinarily large, the inside of the cell, which isn't in contact with the plasma membrane, is going to have a difficult time with getting the nutrients and exchanging its waste. You can picture like an apartment complex. The thing gets bigger and bigger and bigger. All of a sudden, the apartments that are in the center no longer have access to windows. And windows and walls, in this case being the plasma membrane in our example, are where we're going to need to have our nutrients exchanged and our waste exchanged. And so the volume of the cell is going to increase more rapidly than the surface area as the cell gets larger and larger, and that's just based on basic geometry. And so as the cell gets large, it's going to have a smaller surface area to volume ratio. So that means that it has has a larger surface. Here, take a look. Oh, I lost my circles. My point is that as it gets larger, it's going to have a harder time feeding the inside of the cell and allowing nutrients to leave because of the change in the surface area to volume ratios. Certain cells are able to overcome those size restraints by becoming very, very, very long, like a tube. And therefore, the ratio of the surface area to volume is going to maintain the relative high or high enough for the waste exchange and nutrient exchange to still occur. All right, so there should be circles here, but the point is that the radius is the distance from the plasma membrane to the center, right? And the surface area is determined with this pi equation, 4 pi r squared. And what that means is that as the distance to the center doubles, the surface area actually is going to increase more and more. So eventually the surface area to volume ratio, right? So the surface area is going to increase 12 to 50, but the volume is going to increase 4 to 33. So that's a ratio where it was originally at the smaller, at this one level, it was a surface area to volume ratio of 3. As we double that, we end up with the surface area to volume ratio of 1.5. Double that again, surface area to volume ratio of 0.75. So that is basically limiting the amount of waste and gas exchange and nutrient exchange that the cell can perform. And so we have to stay within a reasonable compromise. We can't let the cell get too large or else it won't be able to meet the needs of the internal environment at the very center. Okay, so let's talk about connecting structures right now. We talked previously about how skin cells would be held next to each other, so if you pinch the back of your hand and pulled your skin up, you would not be able to tear off. You'd need an extraordinary amount of pressure to be able to tear some skin off, right? The skin is going to hold on to each other, and that's because we have these specialized type of structures that are called connecting structures, right? Two of these, desmosomes and gap junctions, are found only in animal cells, and plasmodesmata are going to be found only in plant cells. So let's talk about these guys here. So desmosomes are going to be basically like little snaps, if you will. They're regions where cells are studded together, and they're going to be found in regions where tissues are going to be stretched over and over. So the cells are going to need to adhere tightly to one another under the stress of movement. For example, the skin, right, gets pulled a lot, or the intestines, where we have a lot of motion of food particles and digested matter, or the urinary bladder, which is going to get stretched with urine over and over throughout its lifespan. Now, another specialized type of junction is a tight junction, which is a waterproof type of junction. So instead of, we were just previously, when we talked about desmosomes, I compared them to like snaps. Tight junctions are more along the lines of a Ziploc bag. So there's a series of proteins that make that Ziploc region, but it's going to make it that it's waterproof, and there's not going to be any liquid content leaking from one set to the other. So tight junctions are very important, where tubes and sacs have to hold contents without leaking. Another example, again, we're going to be referring back to the skin and the urinary bladder. So the point here is that cells have multiple different types of junctions. So skin cells are going to have both tight junctions and also desmosomes. So they're not specific to one type of cell. Cells can have multiple different types of connective structures. And this is what I was just talking about when I talked about a desmosome. So the desmosome kind of look like a snap. So here we have cell 1, plasma membrane, cell 2, plasma membrane. We have proteins that are linking the two of them together that then go into these anchoring proteins on either side. So cell A is going to have intermediate filaments and anchoring proteins on the internal environment, and then also these linking proteins that interact specifically with cell B's linking proteins, which also has anchoring proteins and intermediate filaments on the inside. So again, that's more of like a snap. It's an anchor in a specific location. Now, tight junctions are very specialized. Like I mentioned, they're more like Ziplocs, right? There's a lot of little proteins that are held right next to each other in these tight junctions that mean that we're going to adhere cell A to cell B without any water ever being able to leak through, right? So tight junctions are going to be waterproof. Last but not least, we have gap junctions. So gap junctions are basically pores between one cell to the other, whereby we can exchange usually information. So it's not going to be nutrients. Usually it's going to be excitatory information that's passed from one cell to the other, but it's passed directly through the connexons. So instead of having to go to the external environment and then get picked up by the neighboring cell, it can go right on through, similar to like passing a message by way of tin can to your neighbor. So these connexon proteins are going to form these pores, whereby information can be relayed from cell A to cell B. And we have a similar type of structure in a plant, except remember the plant cells have those pesky cell walls. So instead of having connexons and pores, we have plasmodesmata, which are going to be a way by which the cell membranes are going to have holes that then go through the cell walls of the adjacent of both that cell and the adjacent cell, and again to be able to connect directly with the cytoplasm of the plant cell next to it. And this is very important, particularly for plant cells, because plant cells have to be able to share water. So if one set of cells go through the water much quickly than the other cells, you can actually have a site of water exchange between cell A and cell B, so that we can make sure that all cells are going to be able to have water to survive. So as I just mentioned, gap junctions, I just referred to them as the tin can phone, right? These have connexins that form connective channels that allow information to be passed back and forth. Generally, the information is passed in the form of hormones, but also nutrients and ions that cells A might have more of and cell B needs, etc., can be shared through these gap junctions. And again, plant cells have very similar type holes, but they have to go through the cell wall, and they're called plasmodesmata. All right, thank you guys so very much for sticking with me through that lecture. I appreciate your time. Happy studying and aloha.